Hey CW Bio, Mr. Kennedy here, and these are your lecture notes on protein synthesis. You know, that whole process where you get to read your DNA and make protein out of it. It's kind of like following a cookbook recipe to bake a cake or, maybe Mr. Kennedy's favorite, chocolate chip cookies. That's what protein synthesis is all about. Following a set of instructions and making something at the end, protein. So before we dive into how this process works, I need to introduce you to a couple of scientists. These men are called or named Beadle and Tatum. Beadle and Tatum experimented with ultraviolet radiation in bacteria many years ago, before you were born, but not me because I'm old. So in this experimentation process, they fired ultraviolet radiation at bacteria to cause random mutations in genes in the DNA of the bacteria. And what they found was that by causing these random mutations and changing a specific gene, that the bacteria could no longer produce a specific protein. Hence the connection. One gene, one enzyme. So for every gene that we possess, according to Beadle and Tatum's work, we're going to end up producing one protein or one enzyme, right? I know that's mind-blowing, but, it was new to them. For us, it's kind of like, okay, yeah, I get it. Now, how does that happen? Well, let's look at protein synthesis and find out. So the process of protein synthesis is really, again, just about like reading a cookbook recipe and making something at the end. And that something is protein, and the recipe is your DNA inside the nucleus of your cell. So the key to protein synthesis, if we want to just put it out there, is you're reading DNA so that you can make protein. Like, that's how it works, okay? The first step in this reading of DNA to make protein is a step called transcription, where the DNA has to be rewritten into a code that the cellular machinery can understand. After transcription, we go through translation, where that code that was written for the cell's cellular machinery so that they can understand it will ultimately be converted into a protein. So let's look at how all this works. As we were thinking about the cookbook recipe that is your DNA, the first thing that I want to make sure we understand is that, well, the recipe is a set of instructions that is kind of written in a special code. That special code is based on the order of the nucleotides in your DNA strand. Remember those things that are the building blocks for DNA? The A's, the T's, the G's, the C's? Well, the order that they come in is actually important. It's like putting letters together to make words in a sentence. So if you mix up the order, then the sentence won't make any sense. Well, the same thing is true of the order of the nucleotides in your DNA. The order of the nucleotides between DNA and RNA has to be rewritten perfectly without making any mistakes, so the cellular machinery will ultimately make the exact protein that we want. Okay? Keep in mind that as we walk through this, I'm going to be talking about different types of nucleic acids. Now, DNA, I've been talking about that a lot here. It has a cousin called RNA, ribonucleic acid, which you also need to be really familiar with. RNA is the workhorse of protein synthesis. DNA is basically the template. RNA, well, without it, there'd be no protein. It's that important. So let me just take a moment to remind you what RNA looks like, okay, or basically what it does. RNA is produced, through a process called transcription, which is very similar to DNA replication. The molecule RNA that's made looks almost like, DNA, except that it's not a twisted double helix. It's a single-stranded molecule, and one of the bases, thymine in DNA, is replaced by a base called uracil in RNA. The sugar in DNA is also slightly different than RNA. RNA has ribonucleic acid or ribose sugar, and DNA is deoxyribose nucleic acid and has a deoxyribose sugar. Okay, so you got to keep those things separate. And again, know that RNA is built from the DNA template. So What kinds of RNA are out there, Mr. Kennedy? Well, let me tell you. There's 3 types that I want you to focus on for this process of protein synthesis. Here they are: mRNA, tRNA, and rRNA. Let me break down what is special about each of these. We'll start with rRNA. I like to call this pirate RNA because it's rRNA, right? Okay, so anyways, rRNA actually stands for ribosomal RNA. So the R in front of RNA is for ribosome. So rRNA is actually the molecule that makes up ribosomes that we learned about, is as a cell structure that makes protein, and like that's where these things come from. So rRNA, okay, makes a ribosome, and ribosomes make protein. Now, it's also thought that this rRNA can play a role in bonding amino acids together during protein synthesis. So not only is it the physical structure of the ribosome, but it actually almost like has little hands that like connects things together. So it's pretty important, pretty special. Next is mRNA. mRNA is messenger RNA. So the M in the front is for the word messenger. Messenger RNA. Messenger RNA is the RNA that actually carries the DNA instructions from the nucleus out into the cytoplasm, where the cellular machinery or ribosomes or rRNA can actually read the message. You see, DNA is locked up tight in the nucleus. It can't ever leave the nucleus. So if we need to get a message from the inside part of the nucleus or DNA out into the cytoplasm, we got to have somebody carry it for us. mRNA is that somebody. mRNA is made by literally like building it off of the DNA molecule, and out it travels from the nucleus to the cytoplasm, where it's picked up by a ribosome and read. The third type is tRNA. Okay, T stands for transfer RNA. So transfer RNA is not transferring a DNA message by any stretch of the imagination. What it's transferring are amino acids that we're going to link together to make our protein strand. So rRNA is the ribosome, mRNA carries the message from the nucleus to the ribosome, and tRNA, well, when the ribosome is reading the mRNA, it's tRNA that's going to deliver the appropriate amino acids. Now, there's one other little thing you need to know about tRNA. There's lots of different types of tRNA. There's up to like 60 different types of tRNA. Now, there's a reason for this, and it has to do with redundancy in our genetic code. And as we kind of go deeper into this, I'll try to, during the translation part, remind you of this redundancy. But what the word redundancy means is there's more than one way to code for a single amino acid. So there's 40 to 60 different types of tRNA to allow for genetic variation in us and still the possibility to produce identical proteins. You may not realize it. You look in the mirror, you look around your house, you see people that look very, very different. They have different hair colors, eye colors, heights, so on and so forth. But here's something that you need to know. I don't care like what your, height is, what your hair color is, what your skin color is, what your heritage is. It honestly doesn't matter. Did you know, like, it doesn't matter if I was born in the United States or someplace in Europe or someplace in Asia or someplace in Africa. It does not matter where I was born. When a person eats a cheeseburger, it honestly does not matter where you were born. You all digest it the exact same way. So you can look very different on the outside, genetic variation, but still produce the exact same proteins as somebody else because of the redundancy in our genetic code. And that variation on the outside while producing identical proteins is only possible because of all these different kinds of tRNA. If tRNA, if there was 20 tRNAs to match up with 20 amino acids to make protein, then honestly, we would all look identical. If we were going to make the same proteins, then we would have to have like the same genes, which means no genetic variation. We'd all be identical. That would be kind of crazy. All right. let's get into this process of making protein. Step 1, transcription. Transcription is the process of reading your DNA to make mRNA, or writing your DNA code in a language that a ribosome can understand. Okay? Remember, mRNA is the vehicle that we need to create here to carry our DNA message out of the nucleus and into the cytoplasm. mRNA is an exact copy of your DNA genes. Main difference here is that, remember, DNA has thymine in it, RNA does not. RNA is single-stranded, it's smaller than DNA, not double-stranded like DNA is, so it can actually leave the nucleus and go out into the cytoplasm, whereas DNA cannot, okay? So how do we go from DNA to mRNA? Well, it's very, very similar to DNA replication, where we're just trying to make copies of DNA. You learned that in a different video. So I'll just remind you of some of the steps. In transcription, one of the first things we have to do is unwind our DNA ladder so that we can expose the bases on the inside and use them as a guide to make our mRNA. So the DNA ladder has to be unwound, then it has to be unzipped. Once it's unwound and unzipped, it'll be opened up for enzymes like RNA polymerase to squeeze in there and start literally reading our DNA base by base. As it reads our DNA base by base, we're going to get what's called complementary base pairing that occurs. Complementary base pairing means that as the RNA polymerase reads an A on your DNA, then it's going to grab a U and stick it on there so that it can start building its mRNA strand. And if it reads a G, then it's going to, the RNA polymerase is going to grab a C and link that on. And it's just going to go down the whole gene this way and convert the DNA code into RNA code, okay? When it does that, the 3 base code in DNA that maybe codes for a single amino acid, or might be analogous to a word in the English language, that triplet code in DNA is rewritten as mRNA, and we don't call them triplets anymore. We call the 3 base code in mRNA a codon. So you're going to hear that word going forward, okay? So here's some things. Again, the DNA is unzipped, 1 gene at a time. Complementary RNA bases pair to your DNA bases. And then once that's all done, the mRNA is going to break away from your DNA strand. The DNA will then zip up and rewind and go about its merry business, and the RNA will prepare to exit the nucleus. Now, I hate to be technical here, but there is another step that I'm just going to throw out there for you. It's called post-transcriptional modification. So sometimes, mistakes in this copying can happen, and so we want to proofread the mRNA before it leaves so that we don't mess up our proteins. There are things called introns and exons that have to be dealt with in our mRNA strand. So that's what post-transcriptional modification is all about. We're going to go through and we're going to cut out the introns, material in the mRNA we don't need, and we're going to splice all our exons together so that we just have one nice, tidy set of instructions on how to make a protein for our ribosomes. This is what it looks like. Okay, so basically you've got our DNA strand. It's been unwound and unzipped. Right in here we would have the enzyme RNA polymerase, and it would be reading 1 DNA base at a time and assembling RNA bases to match up to it. The red strand that you see here, that is a piece of growing mRNA. When the RNA polymerase gets to the end of the gene, this mRNA strand will break off, go through its post-transcriptional modification, leave the nucleus. The DNA will stay behind, rezip, rewind, and be none the wiser that anything has happened, okay? So leaving the nucleus, right? The single strand of mRNA will leave the nucleus, and it will do so without any coiling, but that doesn't mean that it remains unmodified. Remember, there's post-transcriptional modification that goes on. Proofreading to make sure the base pairing was right, introns have to be cut out, and exons, spliced together. All right, so the RNA nucleotides. The RNA nucleotides that are assembled in this process, they are adenine, guanine, cytosine, and uracil. So if you ever see U's in a strand, right, of A's, G's, and C's, then you're looking at RNA. This is just something to remind you of the code, right? In DNA, 3 bases represent 1 amino acid, or what is analogous to a word in the English language. As it's rewritten into RNA, the word triplet goes away and we use the word codon. Okay, codon. Now, here's the post-transcriptional modification. As the mRNA is made, remember that proofreading occurs, and so that you can write it in your notes, here's exons and introns. Exons are the part of the mRNA strand that's going to be read and used to make a protein. It sounds kind of weird to call them exons, because when I first heard this, I thought exon meant X out or remove, but it actually stands for the word expressed. So this is the part of your mRNA that we got to keep, okay? Now, in between exons, we have introns. That's why they're called introns, because they're in between, as opposed to an intron being something you keep in. This is something you got to cut out. Honestly, we don't really know why they're there to begin with. Some people think they're ancient genes that maybe we used to use at some point, but we don't anymore. But there's no way to prove it. So this is just gobbledygook that we got to cut out of our mRNA strand so that our ribosome doesn't have to mess with it and can just make a nice, clean-cut, functional protein for us. That brings us to translation. So translation is the process that occurs when this mRNA strand is read by a ribosome. Okay, so the mRNA strand, it's been made from the DNA template. It's gone through its post-transcriptional modification. It's moving out of the nucleus into the cytoplasm. Once there, has to be picked up by a ribosome in order for translation to occur. So this is the point at which modified, post-transcriptional modification, modified mRNA is read and a protein is made. Super important that we remember the order of bases in mRNA, which was built off of our DNA template. The order of bases is critical to the formation of a functional protein. 3 bases in our DNA strand, called a triplet, led to 3 bases matching up to that in our mRNA strand, now called a codon, and that codon is basically going to code for or call for an amino acid to be delivered to our ribosome. So the sequence of those codons will determine the sequence of the amino acids in the protein. Very, very important. So here we go. mRNA strand in the ribosome. In order for translation to begin, the mRNA strand must be picked up by the ribosome. That ribosome can be free-floating in the cytoplasm of the cell, or it can be attached to the endoplasmic reticulum. Do you remember which type of endoplasmic reticulum? That's right, the rough endoplasmic reticulum. Now, the rRNA, or ribosome, is going to match codons to anticodons as this mRNA strand is read. So the mRNA has the codons on it, the rRNA reads the codons, and then it matches a tRNA anticodon to that mRNA codon. I know, that's a lot of codons, but that's how it works, okay? So the rRNA matches the codon and anticodons and then helps the bonding of the amino acids. Don't worry, I have a picture. I'll show you, okay? Now, the connection between codon and anticodon is complementary. So that means, like, if I have AAA on my mRNA, that rhymes, then the complement to AAA on mRNA is a UUU on my tRNA. That one doesn't rhyme. Okay, so they have to pair up the same way DNA bases paired up, complementary base pairing, A's to T's, G's to C's. RNA bases have to pair up too. The biggest thing you got to remember is that there's no T's in RNA. Outside of that, it's easy peasy lemon squeezy. Don't make it harder than it has to be. Here's some pictures to explain. So down here on the bottom, I've got a piece of mRNA. I know that it's mRNA for a couple of reasons. Number one, as I look at it, I don't see any T's. There's U's in it, so that tells me it's RNA. The fact that it's mRNA is obvious because it is single-stranded, right? That's how I know it's mRNA. Looking up here, these are tRNAs. tRNAs kind of have this squiggly, almost like, I don't know, treble clef, figure 80 kind of look up here at the top. And sticking off the bottom of a tRNA, we have 3 RNA bases. Okay, so the 3 RNA bases you see highlighted here in mRNA, that's a codon, AUG. The 3 bases that hang off the bottom of a tRNA, that's an anticodon, and that's supposed to be matched up to a codon, that it's complementary to. This little white dot that you see out here, that's an amino acid. So the tRNA is carrying an amino acid. It's hooked onto it right there, okay? This white thing is the ribosome, and it's made up of rRNA. rRNA always forms these big three-dimensional globular things, so that's kind of a giveaway that I'm looking at rRNA. Now, if we go back through the process that I just laid out for you, this mRNA was probably made in the nucleus. Not probably, it was. It was made in the nucleus. It went through post-transcriptional modification. It scooted its way out into the cytoplasm, and this ribosome picked it up. Now, this ribosome is going to read each codon, or 3 bases at a time, and then call for an appropriate matching tRNA with an anticodon that matches up this codon. Now, if you think about it, the codon is AUG. So what's the complement in RNA to AUG? A should pair with a U should pair with a A, and G should pair with a C. So it would be UAC. So this ribosome is going to sit right here until a tRNA with a matching UAC for my AUG shows up. Okay? When it shows up, it will bond the tRNA to the mRNA temporarily, just to hold it in place long enough for the ribosome to move to the next codon and read it. So here's that happening. Okay, it moves over one. It reads that codon, repeats the process that it just did a moment ago, brings in another tRNA, attaches it there. Once attached, we've got these 2 amino acids that are now in close proximity to each other. At this point, they will form a peptide bond, or a bond that will lead to the eventual formation of amino acid chain and functional protein, hopefully. So these 2 amino acids will bond to each other. That's what you see going on right here. And when that bond forms, the first amino acid from that first tRNA is left behind, bound to the second amino acid. The tRNA that brought it will leave empty-handed. And really, for all intents and purposes, at this point, that tRNA is useless until it gets that amino acid replaced. How's it going to do that, you ask? Well, it's going to wait for you to eat. And when you eat, and you eat something that has protein in it, and you digest that protein, you break the protein down into its individual amino acids. So you can use those amino acids to make your own proteins. You ever heard the saying, you are what you eat? Well, there you go. You are what you eat, because you break those proteins down and you can use them to make proteins, for yourself. So this process will continue all the way down your mRNA strand until the the RNA presents the ribosome with a stop codon. Okay, so that's what's going on right here. The ribosome is presented with a stop codon. Stop codons are codons that don't code for an amino acid, and so they basically tell the ribosome, protein's done, let go. And there you have it. Now, this amino acid chain isn't really functional protein until it's gone through some post-translational modification. So just like after transcription, we got to clean up what we've made. We got to clean up what we've made here and maybe do some folding and tweaking and, molding to get this thing into like a 3-dimensional shape so that, we've got a piece of functional protein. All right, ladies and gentlemen, that at the end of the day is transcription and translation. That's how our DNA is read. That's how we get a protein from it. I hope, this sinks in and you've learned tons. I'm Mr. Kennedy, and I will see you next time.